Hello, welcome to today's episode of the PlantGENE podcast series. I'm Debarpita a plant geneticist and early career researcher from West Bengal, India, and a current member volunteer for PlantGENE. Today, I will be co-hosting this podcast with Dr. Aimee Malzahn, the coordinator of PlantGENE. Hi, Aimee. Please give a short introduction of yourself and PlantGENE.
Thank you, Debarpita. I'm Aimee Malzahn. I'm a staff scientist at the Boyce Thompson Institute in Ithaca, New York, and I'm the project coordinator for PlantGENE. PlantGENE is a project focused on advancing bioengineering through community engagement, and you can learn more about the project by visiting our website.
Thank you, Aimee. So today we are honored to welcome Professor Kailash Chandra Bansal as our guest on the podcast series. Dr. Bansal is a distinguished plant biotechnologist from India. He has served as the former director of the National Bureau of Plant Genetic Resources, New Delhi. He has also served as vice chair of the Commission on Genetic Resources of FAO United Nations. His lifelong work and expertise focused on plant biotechnology, functional genomics, and transgenics. He served on the editorial board of the Plant Biotechnology Journal and is a current member of the PlantGENE Global Network. We welcome you, sir, and we are honored to have you with us today.
Thank you.
So I'll start with the first question. You've had a long and impactful career within your country and globally as well. Looking back, what inspired you to pursue plant genetic engineering and molecular biology at a time when the field was still emerging in India?
Well, Aimee, you know what? Actually, I did my PhD in plant physiology sometimes in late 80s. And that time, this new area for us in India, plant genetic engineering and molecular biology was just emerging. So I was looking to update my knowledge. We did not have any kind of a degree course that time, like PhD we have in biotechnology. So I was trying to learn about these latest tools and technology for applying them for crop improvement. So I got a break by attending a first training ever conducted, by the International Center on Genetic Engineering and Biotechnology here in New Delhi, 40 days training course, which was actually on molecular biology of chloroplasts. And I was very fortunate to get selected for that training course. And after I completed that training course, which was, I believe, in 1989, I was fortunate, then to go on to Harvard University, in Cambridge in USA to pursue my postdoctorate, with Professor Lawrence Bogored, at the Center of Cellular, and Molecular Biology, Harvard Biolabs. And then I returned to India. I thought I'm now prepared to take this area of plant genetic engineering and molecular biology and to apply it for improving crop plants, particularly for abiotic stress tolerance, because that's really a big problem for us in India when we talk of agriculture, which is mostly, again, rain-fed, some of the crops. So we wanted to improve, abiotic stress tolerance. So I started working on, identifying new genes, studying functional genomics, and then use them for developing transgenic plants. Even I constructed several of those gene constructs and shared with many international or many national labs actually in India, who then later developed transgenics in several crops.
Thanks. That's it's such an exciting time to be at the beginning of plant genetic engineering, and the field is developing so fast. So it's an exciting time to be in this field. And
yes,
yeah, because I, especially too, there's been so many advances, but now with CRISPR, we just see these kind of very transformative technologies in the field. So it's exciting times. And so my next question was your leadership at the National Bureau of Plant Genetic Resources, or just NBPGR for short. You oversaw the modernization of gene banks and the strengthening of plant diversity characterization. What would you consider your proudest contribution from that phase of your career?
Well, that's a good question. When I joined as head of the institution in 2010 of this National Bureau of Plant Genetic Resources, I learned that we are sitting on a gold mine when we talk of the availability of these wealth of plant genetic resources. But to promote the utilization, what we were lacking was, characterization and evaluating them for various traits which they must be carrying, in those genotypes. So I was quite happy, in fact, and must thank my colleagues who supported me to take up a mega experiment on characterization of entire gene bank collections of wheat and chickpea. Wheat, I had an interest because we were looking for some of the traits, related with thermotolerance in wheat, which is very important for a country like India. And so we characterized entire collection of about 22,000 accessions, which were there at that time in our gene bank after removing the duplicates. And we did that mega experiment, which became pretty famous here in India, which was, covered by the Limca Book of Records we have here. And that time, Professor Dr. M. S. Swaminathan visited us, and he was quite appreciative of these efforts. Because the idea I was that time having was to promote the utilization of these plant genetic resources by breeders. So until unless we characterize them, evaluate, identify the suitable donors, for a particular trait, they will not be able to make, really speaking, much use of it. So that was quite, an exciting time for me that we were able to do it. And once I was successful in doing it in wheat, then we jumped on to doing it in chickpea. So these were two crops which were able to, really speaking, make a quite difference and to identify some of those important donors for not only for abiotic stress tolerance or thermotolerance in wheat, for other disease resistance, traits as well in diverse genotypes, including some of the wild species of wheat.
Yeah, very interesting actually. It's what the pioneer work he has done. So, sir, I just want to ask you, how did your global experience? You have worked with FAO and other international collaborations as well. So, how does that shape your vision for the Indian plant science and Indian plant biotechnology?
Actually, when I started going to some of those meetings of FAO, particularly the International Treaty on Plant Genetic Resources for Food and Agriculture, I realized that there's a lot of, avenues there to promote the exchange of germplasm. See, at one time, before CBD came into being in 1993, the exchange of germplasm was easily available. It was free for all, from anywhere we could utilize it. But then later, when these international, legally binding treaty came into being, like ITPGFRA, as we call it, I learned that we promote the exchange of germplasm among international partners. And again, the idea was to promote the utilization of this germplasm, not by Indian scientists, but across the world, because we all actually share, our vision, our, goals of, fighting climate change or malnutrition in some of the, South Asian or African countries. So the idea was, if we can promote the exchange of germplasm by implementing in India being a partner and signatory to international treaty, plant treaty. So I was happy to get that treaty implemented in action, from the approval from Government of India, the national focal point, which is Ministry of Agriculture, and Farmers Welfare in India. So that was a great experience for me of learning, and I was happy to, be working with some of those stalwarts, coming from various countries around the world, and interact with them and learn more about the utilization of plant genetic resources, particularly for climate resilience and sustainable agriculture.
Yeah, so climate change and climate resilience is currently a big focus for all the sectors of plant science, and plant biotechnology is a very advanced stage of that. It's very interesting that how you have worked with it, and you have brought the global experience to the Indian platform also. So my next question will be, many young scientists often feel disillusioned by the regulatory delays, and there are public resistance. The public are not very much accustomed to the concept of genetically modified crops. So what advice would you like to give to them for balancing the innovation and responsibility?
Yeah, that's a good question. In fact, I agree with you that we have little slowed down process of our regulatory approval of some of these important genetically modified crops. And India, unfortunately, after having commercialized BT cotton way back in 2002-3, about 20 years ago, there's no other crop which has been commercialized. And so I can understand, there could be some kind of resentment among particularly the young students who have been engaged in developing these genetically modified crops for the last many, many years. But nevertheless, the message for those young students would be that we all must continue working on some basic aspects, of functional genomics and studying the mechanism of some of the physiological processes. For example, to increase the rate of photosynthesis. Later, if we can identify those genes which could be utilized for carbon capturing capacity or for utilization of nutrients from the soil efficiently by the plants. So in short, what I'm trying to say is that while we are not able to get these, genetically modified crops released into the environment for the cultivation by the farmers, but for the researchers, particularly young researchers, there are a lot of avenues to continue working in this area of molecular biology, which is so exciting today in the whole world, to identify some of those, candidate genes, novel genes and alleles, which could be later utilized even as markers in conventional molecular breeding aspects as well. So there's a lot, one can really continue doing it. But at the same time, yes, we feel we are working in India to streamline further the process of regulation so that we can bring, more transgenics, more GM crops, which are in the regulatory pipeline for commercial cultivation in India for the benefit of farmers.
So I wanted to follow up on the discussion about biosafety and regulation. So as someone who has been closely associated with biosafety, regulation, and gene conservation, how do you view the role of genetic engineering today, particularly in addressing food and climate challenges?
Well, genetic engineering is certainly a wonderful technology. We must utilize it to a fully possible extent, because, that allows us to utilize, the genes which are available in the global gene pool, So that's a wonderful technology. As you know, in conventional plant breeding, you can only have some limited resources of improving a crop plant by transferring the genes from one species like rice to rice or wheat to wheat. But when it comes to utilization of the other kind of a global gene pool, which is available in other organisms as well, like in bacteria, for example, we have resourced Bt gene from bacteria, and that's only possible through the process of genetic engineering. So I think genetic engineering has a long way to go. Already it has given dividends, I would say. Many countries, 30 countries currently growing about 200 million hectares under GM crops, and another 42 countries consuming it. So there are total about 72 countries. And there's a lot of benefit attached to this technology. And it has been proven from time to time that it is quite safe, or as safe as their conventional counterpart crops are. And so therefore, we must continue using genetic engineering, again, for sustainable agriculture and for meeting some of those requirements which are important for the farmers' income growth, as we have seen by reduced pesticide use in our country, particularly with BT cotton.
I absolutely agree with that assessment. Definitely, this is the future, genome editing, and we need to make the regulations, updated with the science. Particularly, it's the research that has been shown that it's safe, and effective for farmers. So I wanted to talk about more open access research. So you supported open access and public sector science very strongly. How can networks like PlantGENE help democratize plant transformation capacity in developing countries?
Well, that's actually speaking very important, Aimee, at this juncture in our times, because global cooperation is very, very important. And particularly, we must thank PlantGENE platform to provide this kind of a networking, particularly for young researchers, to help them with genetic transformation. Genetic transformation, unfortunately, is a bottleneck when you try to develop with more efficiency, more and more transgenics or even genome edited crops. So therefore, it's important that PlantGENE continues to provide that kind of networking, particularly to these young researchers. And there are several species and plants which are important from agricultural perspective, but they're recalcitrant, hard to regenerate. And so therefore, it's important that we innovate some of those technologies with the help of, again, experts we have in the networking of our PlantGENE, expertise available around. So we must try to utilize it fully. And I'm very happy and must thank the great service provided by PlantGENE platform to these researchers through webinars. And, I have also attended some of them, and I was really immensely benefited with those programs.
Great. Yeah, we're happy to have you as a member, and so thankful to everyone who's been a part of the community. And it's always great to hear that the webinars are useful to people, even experienced researchers such as yourself.
Thank you. Yeah.
Sir, I'd like to ask one question that India recently approved the release of a gene-edited rice variety developed by IRI using the SDN1 technology. Someone who has long championed genome editing, what are your views on this development? What opportunities and challenges lie ahead?
Well, this is a breakthrough, I would say, with regard to Indian agriculture. And I must congratulate those scientists who have developed this technology and these genome-edited rice varieties in India. And these two varieties, particularly for rice, are mega varieties, otherwise very popular with the farmers. And these two varieties carry some of those features which are very important from the climate resilience point of view as well, that they have, like high yields, substantially high yields, about 90 to 20%. And, one of the varieties used with the CKX2 gene is early by 20 to 25 days. And it's got reduced water requirement, reduced requirement of fertilizers. And the other variety is also giving about 20 to 25% of advantage under alkaline soil conditions. Particularly also in the salinity soils, as you know, these problems are emerging more and more with climate change. So overall, I would say that these two varieties are important, and we are working further, on multiplication of the seeds, and should be available, I hope, fully soon, to the farmers. And after these two genome-edited rice varieties have been developed by the scientists of Indian Council of Agricultural Research, I would say, we have another mega project by the ICR itself, wherein we are covering 40 crops, and which is in fact being administered, this particular project, in 34 institutes of Indian Council of Agricultural Research, ICAR as we call it, of Government of India. And similarly, we have a council and industrial research institutions, about 10 of them. They're also engaged in some of the aromatic plants, and gene editing is used in a big way in India. And as you said rightly, that we have this opportunity for us for particularly using SDN1 and SDN2 categories, because we got them exempted from Government of India. We must thank Government of India, our regulators, to exempt them from biosafety assessment, because, we are not bringing any foreign gene into the genome-edited varieties, particularly when we talk of SDN1 and SDN2, and there's no major change, as we would expect, in case of GM crops. Only the native genes are modified to a minimal possible extent, and to have these two important varieties, or rice and even other crops we're expecting soon.
So, sir,
lot of opportunities ahead.
Yeah, lots of opportunities. But what do you think, as a plant biotechnologist, what do you think will be the consumer's response to it? Can you give what your predictions are?
No, consumer response will not be no different than the conventionally bred crops, because I just said there is no biosafety related issues with genome editing at all. In many of the countries, more than 30 countries have already declared, without the presence of any foreign gene or exogenously introduced gene, these crops are considered as good as conventional bred crops. Or even these genetic variation which we are introducing through genome editing, they're also happening in nature, naturally occurring, mutations. So therefore, there's no concern at all with regard to biosafety, and accordingly, I'm very confident that consumers will be happy to accept it, and more so, even farmers would be very happy to grow them, because it's going to save them a lot of labor and time and give them better income.
Okay, that's very encouraging. And also, as an early career researcher from India, I'd like to ask you that many of the young people who are in this research section in India and other Asian countries, they have immense capabilities, but they lack the lab exposures, training, and international experiences. What is your message to them?
Well, my message to them would be that they should find, actually speaking, their own way by working hard. They must, identify the collaborators, not only outside India, even within India, there are several labs where can get the basic training. If my own experience, I got that basic training from within India. And then I had an opportunity, to go outside, as I mentioned before, to Harvard University. So similarly, what I'm saying is there are very many opportunities today, rather in this world of internet, when we are so well connected through webinars and by, some of these platforms like LinkedIn, they provide you wonderful opportunities. But it's very important. They must, be focused, have their skill developed in this particular area of research. As a scientist, they must go deeper into those questions of science, and I'm sure, many of those international researchers will be very willing, to partner with them, to give them an opportunity to come and work with them. Funding opportunities are also plenty available if you go around. So I would encourage them to really speaking, be, kind of on the watch out of these kind of possibilities, available, and they must be willingly, in a positive frame of mind, contacting people all around. I'm sure sooner or later they will get a break. They should not get disheartened if, they don't hear anything immediately. So there are a lot of opportunities today, even more than what we had in our times for these youngsters.
Yeah. So thank you so much for the encouraging words. It's really helpful.
So we've talked earlier about biosafety and regulation, but also that this field around transgenics, and then now we have CRISPR technology on the market. There's been just a rapid growth in the biotech field. How can we balance between innovation speed and biosafety assurance in the context of CRISPR-edited crops?
See, as I said before, Aimee, with regard to CRISPR genome-edited crops, at least in India, as per our Government of India regulatory framework, we have no biosafety concerns attached to it at all. We are, at a stage, though we have regulation, of course, I must say that, but only at the initial level. We call it as our approval by the Institute Biosafety Committee. But in case of GM crops, there are two more tiers where get subjected to the biosafety regulation. We have got one review committee on genetic manipulation, followed by a genetic engineering appraisal committee. That's for the GM crops. But for genome-edited crops, since we have declared them as equivalent to non-GMOs, because there is an absence of foreign DNA in the final product, we are not attaching any kind of a biosafety-related concerns with it at all. So therefore, we are trying to move with the speed. This is a wonderful, innovative, transformative technology, as we all know about it. And this is the era we are passing through. We are fortunate, actually, and we must, thank these two wonderful scientists and Nobel laureate, both of them. They came up with this technology, and it's like a boon, I would say. For some of the countries like India, where we need more of these crops for food security, nutrition security, and even for climate resilience, it's so very important. So this allows us to move with the speed. Only thing is we must try to collaborate with each other, and we must try to identify those target genes, which are very important, in a particular crop, particular variety, which is popular with the farmers, which we are trying to go for genome editing. So it's a wonderful technology. And I don't think there are any biosafety concerns. There should not be, scientifically speaking, even anywhere around the world.
Yes, I agree. And, you mentioned something earlier that transformation is really the bottleneck to working with these crops. So it's not the CRISPR necessarily that, although that can be a difficult process, but really the transformation, getting your CRISPR construct into these plants is really challenging. So I hope to see those methods and that technology advance.
Right, but a lot of good research is also being done, by developing some of those tissue culture-free, genotype independent systems and protocols through virus-mediated delivery of some of those CRISPR reagents.
Yes, it's been exciting to see that technology develop. And a little bit of a different question. So you've been a strong advocate for building research networks, one of those being the PlantGENE community. How important do you think peer-to-peer collaboration is in today's science, especially across borders?
Well, I think it is more important than ever before, because as we are, experiencing more and more changes related to climate change, I think we all have a shared vision. We all have a shared goal to have, a sustainable way of food security. and also nutrition security in some of the countries, like I mentioned before, in South Asia, including India and African countries. So I believe peer-to-peer collaboration, it's a global world, it's a global village, where we can try to help humanity at large, through our scientists, kind of discoveries. And I'm of the firm opinion, that as far as food security is concerned, food is concerned for the person who is quite in need of it. Anyone should not be dying out of hunger today in this 21st century. So I think we all scientists, irrespective of our origin of the country, if we have that expertise, and as I said before, that PlantGENE is a wonderful platform. We should bring all those like-minded scientists together, irrespective of their country and all, origin, and we should work for those countries where the food is important, and food is need to be produced more, and that too with quality included into it. So I think it's important, and irrespective that how much contribution we make in science, but it will be only measured if the needy person who is unreached today, with regard to if we reach him or her, through our scientific innovations, will be a better, kind of way of providing and contributing to the world through science. So altogether, I mean, no single person probably can achieve that. I think we must all work together, and I appreciate your concern. You mentioned about peer-to-peer, collaboration is very, very important in a very positive fashion. We can change the whole world.
I love that. That is such a positive outlook. And I think that's, I was going to say everyone, but maybe I shouldn't speak for everyone. But I think so many of us got into this field to make a difference and to address these issues around climate change and hunger. And it's fantastic to be in a field and be with like-minded people that are working towards this goal.
Yeah.
So that is we are out of time today, but I just want to thank you so much for such an interesting discussion and just, a look into all the incredible research you've accomplished during your career so far. And I really appreciate the insights as well as the look forward to what we can do to address these global issues. So thank you.
Sure. Thank you very much.
Thank you, sir. Thank you very much for joining us today.
It's a pleasure.
PlantGENE would like to thank the National Science Foundation for their support. To learn more, please visit their website at nsf.gov.